Hello friends, welcome back in the video session of pharmaceutical analysis. So in today's session we will be discussing very important concept, that is quantitative applications of fluorescence spectroscopy and choice of fluorimetry over the UV visible spectroscopy or the absorbance spectroscopy. So we have seen the principle and theory of fluorescence spectroscopy. We have seen what is the difference between fluorescence and phosphorescence, and this is basically an quantitative spectroscopic technique. So we can find out the concentration of analyte in the given sample. So we will be discussing the applications of fluorescence spectroscopy with main emphasis on the pharmaceutical applications. So the applications of fluorescence spectroscopy are almost as wide as one of the imagination. So we have restricted our discussion in this particular session only to few examples in the particular area of this applications of fluorescence spectroscopy. So it is very hard to imagine the chemical and biological science without this particular technique nowadays, because of the modernization of sophistication in the liquid chromatography and even in the instrumentation of fluorescence spectroscopy. So this fluorescence spectroscopy is one of the most widely used molecular spectroscopic technique in the field of molecular biology, biophysics, biochemistry and the pharmaceuticals. So these are the fields where we can have wide varieties of applications of this particular spectroscopy, and the main reason behind using this spectroscopy is its selectivity and sensitivity. So this is very sensitive, very selective method, and because of that the method is very widely used. And some important applications we have already outlined in the previous discussion. So we'll see one by one what are the applications of this particular technique. So the first application of this fluorescence spectroscopy is in the food industry. Now we know nowadays the quality of food and the standards are also stringent, and we need to check the food material for its quality, whether there are adulterants or contaminants present, and whatever the components of a particular food material, in what concentration they are present. Are they present or not? If they are present, in what concentration they are present in the foodstuffs. So this fluorescence spectroscopy plays a very important role or function in the modern food analysis, as can be seen from its wide use in the determination of numerous food components and the contaminants present in it or the additives which are added in that, and most importantly we can quantify, we can determine the presence of adulterants along with their amount. So this technique has made available very sensitive and selective method that will satisfy the requirement of food analysis, because the method is very selective, it is very specific method, right? So selectively we can quantify, we can identify a particular analyte or the contaminants present in this food material, right, which are usually very complex. So we know in food material there are a lot of components or the materials which are present in one particular food packet or the food stuff. So the nature of the sample of food is very complex. So in presence of all other contaminants or the components, we need a method which selectively identify and quantify a particular analyte. So this can be done with the help of fluorescence spectroscopy, taking the chromatography for the separation. Okay, and we can very frequently determine the concentration of these contaminants, additives or adulterants at very low concentration, okay, in the wide varieties of foodstuffs which are available in the market. So initially in the previous days this fluorescence spectroscopy was restricted only to the species or the samples which are fluorescent in nature. So they have inherent fluorescent property, they have the conjugation in their structure. So the foodstuffs only are able to show the fluorescence, only those kind of substances were analyzed in the earlier days. But because of the modernization and the availability of some methods which converts the fluorescent the non-fluorescent substance into a fluorescent substance. So we have the physicochemical means or the chemical methods or the photochemical methods which convert the non-fluorescent substance into a derivatized substance which becomes fluorescent in nature. So nowadays we can also extend the fluorescence spectroscopy with the non-fluorescent substances also. We will convert into a complex with the help of some chemical reagents, and with them we will convert the non-fluorescent substance into a fluorescent substance. So there are numerous techniques involved in this fluorescence detection, and usually we take help of liquid chromatography, okay, or even nowadays the capillary electrophoresis is also used, and these are the separation techniques. So these complex kind of substances or the foodstuffs or even the pharmaceuticals, we can initially purify them, right, and once we purify, the purified components will be subjected to the fluorescence spectroscopy for better accuracy and the precision. So in a broad way we can summarize here, the fluorescence spectroscopy can be used in the determination of vitamins, proteins, peptides, amino acids and amines, and also to quantify the inorganic species which are present in the foodstuff. Then the determination of adulteration, usually it is very common nowadays, so we can find out the adulterants present in the foodstuff, in what concentration they are present, and ultimately by quantifying these adulterants we can check or we can judge the quality of the food material. Then we can also find out the amount of food contaminants like polycyclic aromatic hydrocarbons, mycotoxins, phycotoxins, nitrite, nitrate and nitrosamines, pesticide residues, veterinary drug residues, microbial contamination and the food additives. So all these contaminants which are very commonly present in the foodstuffs can be easily quantified with the fluorescence spectroscopy. And the non-fluorescent substances present in the pharmaceuticals or the foodstuffs can be chemically derivatized, and the derivatized form will be fluorescent and they will be quantified with the fluorescence spectroscopy. The fluorescence spectroscopy also has or find applications in the field of agriculture, right. So for instance, you can see the identification of different crop varieties, and these different crop varieties we can use or we can find out with the help of fluorescence spectroscopy. So laser induced fluorescent emission technique, which is abbreviated as LIFS, sorry. So this is an excellent tool which is used to identify the citrus seeding varieties. So you know the citrus fruits, and these citrus seeding varieties will have different amount of the citrus component present in these foodstuffs. So we can quantify that, and then based on the quantities we can categorize them into a different varieties. So this is one of the application. Likewise there are many applications you can use it in the agriculture industry. So we are discussing with the main emphasis on the pharmaceuticals and the foodstuff. So I have listed only one example in the agriculture. Then applications in the environmental monitoring. So in this environmental monitoring, the technique also has wide applications. So one of the example we can take here in the treatment of water surrounding the landfill areas. So you know, when the rainwater is usually percolates through the waste and liquid forms during the biodegradation of waste in the landfill, and this landfill leaches the uh, contaminants, right. So this leachate contains a mixture of pollutants that will be harmful to the environment, right. So the rainwater is percolating through these solid waste, and then those waste will leachate in the rainwater. So we can find out the amount of these impurities in the form of leachates present in the rainwater, which is harmful to the environment. So we can use a fluorescence spectroscopy, we can take that water as a sample and we can quantify those leachates which are usually fluorescent in nature, okay. So this is one of the application in the environmental monitoring. Then we can extend this spectroscopic technique even to the bioscience. So bioscience is again a major field where we find applications of fluorescence spectroscopy, and one of the most frequently used application of this technique is in the detection in the quantification of DNA and RNA with the high precision. So you know the precision is the uh, repeatability of the result, right? So with high precision we can quantify the DNA and RNA present in the biological sample. So what usually we do, we take an extrinsic fluorophore, often the ethidium bromide. So this ethidium bromide is a chemical reagent which is usually having the fluorophore, and this is reacted or added with the DNA or RNA sample. So after reacting this with the DNA, so it will convert the DNA into a complex of DNA with the fluorophore, and then that will have a very good fluorescent property, and then we can easily find out the concentration of the DNA or RNA in the sample. Okay, so this is one example. Now the most important is in the applications of pharmaceuticals. So this spectrofluorimetric technique are also used widely in pharmaceuticals to analyze mainly the drugs present in these pharmaceuticals. So an example is the analysis of co-formulated tablet prescribed as cholesterol medication. Okay, so you can see the synchronous fluorescence spectroscopy usually provides a simple, it is very fast and accurate method for analyzing a tablet called Atoriza. Now this Atoriza tablet usually contains a combination of two drugs, that is ezetimibe and atorvastatin calcium. So these two drugs can be very easily analyzed by the fluorescence spectroscopy, and this is one of the ideal method for routine quality control check which is used for this particular medication. Even we find in some of the official monographs the fluorescence spectroscopy is listed for determination of percentage purity of such type of formulations. We can also have the applications of this fluorescence spectroscopy in the clinical samples. So clinical and drug application by using chromatography and then subjecting to the fluorescence spectroscopy. Usually we know the drugs, when we take it inside the body, they will be going into a biological fluid, and then you know biological fluid is very complex in nature. For example, you can see the amino acid. So there are various amino acids. For example, you can consider the tryptophan. Now if you want to quantify the tryptophan which is available in the biological fluid, now let's say if I want to quantify the tryptophan in the brain tissue or in the plasma or in the urine, so this biological sample I will take which contains the tryptophan, and this biological sample I will purify with the liquid chromatography. So you know liquid chromatography is a powerful separation technique which will separate out the mixture into individual fractions. Now once you separate the complex mixture into individual fraction, that means you are purifying the sample. Now once you get a pure tryptophan from this biological sample, then we can subject it for the fluorescence spectroscopy. So in this case, separation with the help of chromatography and then separated fractions will be subjected to the fluorescence spectroscopy for better accuracy. So that is possible, and in many clinical areas we find the fluorescence spectroscopy is often combined with the liquid chromatography. Then we can extend it for number of categories of drugs like analgesic, antineoplastic, anticonvulsant and so on. I have listed here only three of them. Like analgesic, you have ibuprofen. So you can quantify the amount of ibuprofen present in the plasma, amount of naproxen present in the serum. Then antineoplastic, you can see the daunorubicin, doxorubicin present in the plasma, urine. So you can take these biological samples or the clinical samples, subject it to the liquid chromatography, get them purified, and then subject it to the fluorescence spectroscopy for better accuracy, and we can quantify with great accuracy and precision because of the sensitivity and specificity of the method. Then how about extending this particular technique for the non-fluorescent substances. So there are certain substances even in pharmaceuticals which do not have conjugation in their structure, which are not able to absorb the UV or visible light, and in turn they will not show the fluorescence. So we can convert them into a fluorescent substance by one of the popularly used method. We refer it as a chemical derivatization. So by taking some chemical reagents, we can convert the non-fluorescent substance into a fluorescent substance. So one such popularly used reagent is the dancyl chloride. Now this dancyl chloride has a chemical name 5-dimethylamino naphthalene 1-sulfonyl chloride, which is popularly referred as a dancyl chloride. Similarly, you have dancyl iodide and so on, dancyl bromide and so on. So this is the reagent that react especially with the primary amines in both aliphatic and aromatic kind of structures, and this will usually produce a blue or blue-green stable fluorescent sulfonamide adduct or the complex, and this can also react with the secondary amines also. So what we are doing, we are taking a dancyl chloride which is capable to react with the primary or secondary amine of aliphatic or aromatic compounds, and we have a lot of pharmaceuticals which will be having in their structure as a primary or secondary amine, and then we can convert them into a fluorescent substance. So one such example I have listed here for your understanding. You know the barbiturates. Now here I have drawn the structure of barbituric acid. Here you can see it has a secondary amine. So this NH is a secondary in nature, even this NH is also secondary in nature. Now in this case the dancyl chloride, so this is a structure of dancyl chloride. You can see there is a naphthalene ring, and at fifth position there are two methyl groups attached to this particular amino group, and at one position there is a sulfonyl chloride. So this SO2Cl, we refer it as a sulfonyl chloride. Now this sulfonyl chloride will react with the primary or secondary amine. So in this case it can react with this NH, or even it can react with this NH, and then it will form the corresponding complex or the adduct. So we refer this as a dancyl derivative of barbituric acid. So you can see here the chloride has been removed along with the one of the hydrogen of this nitrogen, and then you can see this nitrogen of the barbituric acid is getting combined with this, forming a dancyl derivative of barbituric acid, and this derivative is colored and fluorescent in nature, whereas the barbituric acid or the barbiturates are non-fluorescent. So this is how by chemical derivatization, this is referred as a chemical derivatization. So by taking some of the chemicals, for example here dancyl chloride, we have converted the non-fluorescent substance into fluorescent substance, and then this fluorescent substance can be quantified with very ease with the fluorescence spectroscopy. So this is basically an dye, dancyl chloride is the dye. So you can extend this particular technique to number of pharmaceuticals which are non-fluorescent. Some of them I have listed here, as you can see, the maprotilene, then prebexylene, right, then barbiturates, cannabinoids and so on, and you can also quantify them in a clinical sample or the biological sample, and this is the reaction which I have represented as a pre-reaction, right. So that means you can directly take those samples, and before uploading into a column only you can have a chemical derivatization. Then next is fluorescamine. Okay, so this fluorescamine is a spiro compound. You can see in the structure, right. So the two rings which are fused with one single carbon, we refer it as a spiro compound. We have studied in this bridge head compounds, right, the fused kind of heterocycles. Now in this case this is combined with the one carbon, two rings are combined with the one carbon. So this is a spiro compound, and this compound itself is non-fluorescent, but when it reacts with the primary amine it will form a highly fluorescent product. In earlier case the dancyl chloride was fluorescent, right, and it was forming a fluorescent substance, but in this case the substance is non-fluorescent, but when it combines with the non-fluorescent substance having primary or secondary amine, it will form a fluorescent substance. So this is used as a reagent for detection of amines and peptides. I can see this particular reagent combine here with the amine and it will form the complex which is fluorescent in nature. Okay, so you can see there is a ring opening here, so this bond between the carbon and oxygen is getting broken, and then this will be a carboxylic acid here, COOH, and this oxygen is substituted by the nitrogen of the amine, and this is what is getting formed here. So initially both were non-fluorescent, but after combining them or by the chemical derivatization, the formed product is fluorescent, highly fluorescent, and then you can easily quantify with the fluorescence spectroscopy. So one such example you can see here, the amphetamine. Now this amphetamine can be easily reacted here with this reagent, and then we get the corresponding adduct or the complex. So you can see the ring has opened here, it is forming a carboxylic acid, and the nitrogen has substituted the oxygen of this particular spiro ring, and then the fluorescent compound has formed which you can quantify. So similarly you can see the other drugs, that is the cephadrine, amphetamine and so on. So you can also determine them in a clinical samples. If you are determining in a clinical sample, in this case the reaction is post reaction. That means you take this clinical sample, separate them in a chromatographic system, and then after separation you derivatize them and then subject it to the fluorescence spectroscopy. The third is the orthothalaldehyde. So this is another popularly used reagent which is also referred as a thalaldehyde or orthothalaldehyde, or it is abbreviated as OPA. So this OPA is a chemical compound with the formula C6H4CHO outside bracket 2, and you can see here the benzene ring and CHO is there, aldehyde group is there. So this is one of the 3 isomer of benzene dicarbaldehyde. So it reacts with primary amines, all primary amines containing analytes or the pharmaceuticals, and that will yield the fluorescent isoindole derivative. So this is forming an isoindole ring, right, and this becomes highly fluorescent in nature. So you can see the OPA has reacted with methionine and lysine as an amino acid, and then it has formed a corresponding adduct or the complex which is having a blue fluorescent color. So this you can easily quantify with the fluorescence spectroscopy. So earlier these two were non-fluorescent, even OPA is also non-fluorescent, but after reacting with this it becomes, the product becomes the fluorescent, the complex or adduct becomes the fluorescent. So this is again a pre-reaction. So before loading into a column, if you are applying with the biological samples in the form of plasma, urine and so on, so you can have this derivatization and then you can subject it to the column. So the drugs which have the primary amino functionalities like gentamicin, histamine, amphetamine and so on, that can be analyzed, quantified by this particular technique. So these are the applications of fluorescence spectroscopy with the pharmaceuticals and other fields, right, and even as I mentioned, we can extend it even to the non-fluorescent substance, especially by using the chemical derivatization. Now next is the choice of fluorimetry over the UV visible spectroscopy. So how we can select which method is better for my particular sample? So you know, although measuring the fluorescence may not be common as measuring the absorbance, because UV visible spectroscopy is very widely used and that is very common, but the fluorescence spectroscopy is not so common. So it may be even more powerful method for the sample characterization. So it is important to understand the strengths and weaknesses of this particular method which is applying to the different kind of samples. The beauty of this method is its selectivity and sensitivity. The fluorimetry method is more sensitive, more selective as compared to the UV visible spectroscopy. So when we talk about the sensitivity of the fluorescence detection is approximately 1000 times greater than the absorption spectroscopic method. So that is why we consider the fluorimetry is more sensitive method for the quantification with respect to the UV visible spectroscopy. So this leads to the greater limit of detection, obviously, right? The limit of detection will increase with the fluorimetric methods, and when you have very less amount of sample, especially the biological samples like in the form of serum or in the form of urine, where the large amount of sample you cannot withdraw from the patient. So in those case, even with the little amount of sample, because of the greater limit of detection, we can easily quantify those samples with the fluorimetry. And this is important especially when you are working with the precious or limited quantity material, like as I mentioned, the biological samples, or during the research, right? Usually we have very limited number of amount of sample, so we have to select the sensitive methods for estimation. Then next is the specificity. Now here the basic principle of the fluorimetry we have discussed, and only the compounds which are having the property of showing fluorescence or phosphorescence, only those will be detected in the instrument, right? Unless and until you are not converting them into the derivatives. So here only the molecules that is having this particular emission radiation properties, only those molecules will be detected. So you can imagine you have one sample which you are estimating or quantifying with the UV visible spectroscopy. The same sample is capable to produce the fluorescence you are estimating with the fluorimetry or fluorescence spectroscopy. So the analyte, your main analyte is fluorescent in nature, right? So if you are analyzing with the UV visible spectroscopy, along with that analyte, if some other impurities are there which are also absorbing the light, then they will also contribute to the final absorbance. But in the case of fluorimetry, only the fluorescent substance will contribute to the readings, right? In this case, we are measuring the intensity of emitted radiation. So the impurities which are non-fluorescent will not affect your reading in fluorimetry, in short, right? If you have impurities which are non-fluorescent in the sample, they will not be detected, they will not interfere usually in the detection of your sample. Whereas in case of UV visible spectroscopy, they do interfere in the absorption if they are able to absorb the light in the wavelength which you have selected for measuring the absorbance. So obviously because of the sensitivity and the specificity, the fluorimetric method are more accurate, that is very obvious, right? And because of this, these are preferred methods, especially when have limited number of sample, the quantity of the sample is very less, especially the biological samples or the precious samples with the limited quantity. Okay, so based on what kind of sample you have, to what extent you want to detect, right? The limit of detection, all those things you need to keep in your mind when you are selecting between the fluorimetry and the UV visible spectroscopy. So each and every method also have some shortfalls or drawbacks. So there are also certain disadvantages of this fluorimetry in terms of the cost effective way. So when we talk about the economic value of this particular analytical method, the price of the instrument we need to consider, right? And this is one of the challenge to the analyst to complete the analysis in the cost effective way. So here the light source is playing an important role. It should emit the radiations of equal intensity, continuous kind of radiation should be emitted, and here the filters are also playing an important role. And we know there are two filters available here in the fluorimeter, right? One before the sample, which is usually filtering the radiations required for excitation of your sample, and the second filter which filters the emitted radiation which you can quantify, right? So those things are important. So usually the cost of the fluorimeter or the spectropluorimeter is usually high as compared to the absorption spectroscopic instruments, but they're sensitive, they're selective. So what kind of samples you want to find out, with what accuracy you want, with what precision you want, with what limit of detection you want to quantify, all those things really matter when you are deciding between the UV visible spectroscopy and the fluorimetry or fluorescence spectroscopy. Then impurities. So as I mentioned, if the impurities are non-fluorescent and soluble in the sample, they will not affect your final readings. Though they are present, they will not interfere with the final readings, but the impurities are uh, as a particulate matter, right? So there is a possibility that they may scatter the light of the excitation wavelength, the incident wavelength, and because of that they will alter the final readings. So if impurities are present in your sample, if they are non-fluorescent, they will not have any effect, but if they are as a particulate matter, they may scatter the radiations, and that will create the problem in your final reading. So that is one of the drawback here, if the impurities are present as an interfering substance which are scattering the radiations. And then most important is the pH of the sample solution. So we have seen, when we have discussed various factors affecting the fluorescence intensity, the pH is one of the major factor which affects the fluorescence intensity. So at what pH you have prepared the solution, that will create a different spectral properties here. So this fluorescence can be affected by the pH of the sample solution, and this will lead to the inaccurate measurement in the method. So using the similar conditions, maintaining the pH which is desirable for the readings, right, we can maintain that pH by using a buffer solutions or adjusting the pH of the sample solution appropriately. We can overcome these drawbacks, and with that the fluorescence spectroscopy, we can use it to a great extent. It has already been used in a great extent in the industries, especially the biopharmaceutical industries, and even the various drugs and other fields. So that is all about the applications of fluorescence spectroscopy and how we can decide the fluorescence spectroscopy over the UV visible spectroscopy. Thank you for watching it.